Okay, hello everyone. So welcome to the YNOW, first YNOW of this year. So happy New Year to everyone. So today we'll have Ikbal and Bin presenting Biomedical Optics. So I'm really looking forward to this. And for all of you who are new to this seminar series, we have a mailing list that you can subscribe to. You can find it online, and so you can stay in touch with the latest seminars. With that, I leave the floor to Ikbal and Ben. Okay.
Hello, everyone, and thank you for the introduction, and thank you for the invitation as well. It's my pleasure to be presenting you in the first Winehouse seminar series of the year. We are going to introduce biomedical optics to you today with my colleague Bin Deng. I'm an instructor in radiology, and I am in the optics and at Martinos team. Can we move forward?
Yeah, hold on.
So our talk today will be two-part. In the first part, I will introduce you the mostly preclinical applications and microscopy and macroscopy methods. And in the second part, Bin will cover the clinical translated optical tools that we are experienced in optics at Martinos. So before we dive into why and how, I would like to start with a more basic question. What is exactly biomedical optics? Can you move forward? Yeah. Optics is study of behavior and properties of light. So starting from generating it, how we propagate, how it interacts with matters, and especially with tissue in our case, and how we detect it. So when we talk about light, let's define what is light. Light is electromagnetic radiation that can be detected by human eye, and it shows both wave and particle characteristics. So when photon, we keep mentioning it also as photonics. Photon is the smallest discrete amount, or the quantum of electromagnetic radiation. So it is not continuous energy. So it's like discrete energy packets, and it doesn't have a mass, and it travels with speed of light, something around 300 million meters per second. So photonics is like electronics. So photonics is the science that study photons, which are particles of light. So biophotonics, as the name suggests, is actually especially the relation of photons with biological tissues. And overall, the broad biomedical optics field is aiming to design specific applications and optic research to answer the needs of biological or clinical needs. So biomedical optics is a broad field. So it is impossible to cover in an hour, obviously, but we can provide a bird's eye view. So two major categories that we can see biomedical optics are, so we can use light to modulate tissue or apply some therapies, like you see lightsaber. We can burn, ablate, cut. So these are not the category that we are going to talk about today. Or we can use light to do imaging and to do diagnostic reasons. So today we will focus on this category, and we will, in my part, I will mention mostly the preclinical studies, because microscopy applications, most of them did not translate to human use. So our objects are, samples are most of the time cell cultures, organoids, and animals like rodents and others. And in small portion, we also have some extent to human as well. And in the second part, Bin will mention some of the tools like diffuse optical tomography, near-infrared spectroscopy, and DCS, like these are more like clinical monitoring tools. So we mentioned light is electromagnetic radiation. So if we look where we are in the electromagnetic spectrum, we correspond somewhere around the 3 like 200, 400 nanometer to somewhere around 800 nanometer. But as we microscopists, we like to extend this up to 1 micron. Like we love to use that near infrared region for measurements in tissue because it has some special characteristics. So if you, yep, thank you. So we mentioned that light has wave characteristics. So like any wave, it has a propagation direction, it has a wavelength, it has a frequency. So it has all those characteristics. And if you look into the optical window, the portion that we can see our eyes are sensitive to is corresponding to a very narrow band. And in this spectrum, as you go right, meaning increasing the wavelength, the energy of the light actually lowers. And as you go left, it increases, where the frequency also increase, but wavelength decrease. So on the bottom right, you see the Einstein-Planck relation. It actually mentions, explains the photon energy. It is proportional to the frequency of light and also the Planck constant. Thank you. So where does in vivo optical imaging stand in terms of spatial resolution, temporal resolution, and imaging depth compared to some clinical tools that we are familiar? So we, by using optical microscopy tools, we can go down to microsecond range, temporal resolution-wise. It can be very fast or moderately small. It depends very much to the modality that you use. And spatial resolution-wise, it's also provide a broad spectrum. We can go down to sub-micron range, around 0.5 micron easily, or it can be somewhere around centimeter range as well, depending on the different modalities. We will touch to some of them and how it compares to PET and fMRI. It can give a sense, and ultrasound as well. So what are the advantages of optical microscopy? Why would someone want to use optical microscopy for research or clinical use? So some clear advantages of it. So it is safe. It is non-ionizing radiation using optical light. And it has superior spatial and temporal resolution depending on the modality. And it can provide you various contrasts to measure different parameters that it would be very interesting biomedically. You can measure blood flow, cellular or vascular morphology, cell, like you can monitor their movement, the voltage, membrane voltage, and others, and many others. So it is very useful in many ways. What are the limitations of this tool? You can tell, like we cannot see through our hands or we cannot see through our body. So it is not transparent to optical wavelengths, right? So there's a limited penetration depth. So it is most of the time confined to millimeter roughly. And the reason is most of the, like, we cannot go deeper because blood absorbs a lot and water as well. And there's a lot of scattering because of the structure. So because of this limitation of depth, the translation of these tools into clinic has been limited. So we can use it in fairly transparent organs like eye, or accessible like skin, teeth, or when there's a surgical opening, there's an access for optical imaging, so we can use that. Or the cavities like endoscopic application, we can also use that. So let's see the different type of tissue light interactions. How does light interact with tissue? So it can reflect from tissue, it can transmit through, and it can refract. Most of these should be familiar to many of you from high school optics lesson, right? Or it can, if it is a granular surface, it can diffuse reflect. So each of these are critical because these are actually source of contrast for optical imaging. So scattering is an interesting one. So when light interacts with tissue, it can either elastically scatter, we call them Rayleigh scattering. So there is no energy lost when photon is interacting with the tissue, or it can be inelastic. It can either increase in energy, interestingly, like anti-Stokes shifts, or decrease in energy. And we tell them like Raman scattering type. There is no resonance required in this category. So if the photon, when it is interacting with the tissue, is the energy of the photon is resonant, matching to the energy gap that the molecule has, the absorption happens. So this absorption can be followed with a luminescent event or not. So that's also a possibility. So absorption is very interesting to use. So on the right, you see most of the proteins, molecules, oxygenated hemoglobin, deoxygenated hemoglobin, and water, the absorption spectra. So there is a nice shaded window here, which, I mean, water is very important. It is very absorbent. Absorption is high at certain wave. So we need to operate within the band that the absorption is low, so we can penetrate deeper, obviously. So you can see actually there's a nice near infrared window where we love to do our imaging in vivo imaging cases. So we mentioned absorption can be followed by luminescence events. Two most common ones are fluorescence and phosphorescence. So the photon is absorbed, there's some vibrational relaxation, and then the molecule emits a lower energy photon with different wavelength. And that can happen very quickly in femtosecond range, which will represent as fluorescence, or it can experience re-intersystem crossing and then a slower relaxation process, and it can emit a lower energy photon, and phosphorescence can happen, which usually happens around microsecond lifetime range. So by using either of these interaction, this can serve as our contrast mechanism. So we can either use the absorption, scattering, reflectance, or one of these luminescence. We can either use intensity itself or how fast or slow it is decaying, like the lifetime of it, as our contrast mechanism. Can we move forward, please? So can we get away? So if absorption and scattering is so much troubling, is there a way to get rid of them? Yes and no. I mean, if you're working post-mortem, there's actually a very cool method, like over recent years, developed optical clearing methods. So you can have your whole brain. These are, you see some examples, these mouse brains, these are. You can clear the parts that you don't, that's causing you trouble. So you can keep them all together without slicing. You can keep them in these solutions. There are different recipes, many of them actually. It can last like a few weeks or a few days. So you can, at the end, have a transparent whole brain. That's very common now. And labeling strategies are also very developed. So if it is a post-mortem study, you can clear your tissue and then image post-mortem. That's one possibility. One more, please click. Yeah, and it is actually even progressed towards bigger tissues, bigger organs. These are some human organs, actually, some embryo parts. So it is progressing very impressively. So that's one option. Let's keep this as a note, side note to somewhere. So if you're not working with post-mortem tissue, what's going to happen? So can we move forward? So we can use, let's talk about what we can do with the microscopes that we have. So what are the critical parameters that you have? And once you have a microscope, what are the important aspects of it? What should we pay attention to? So first thing, very important thing, is numerical aperture, which is actually how much the capability of, like, light collection capability of your objective lens or of your microscope. So bigger that cone of angle, more light you can collect. So most of the time this is referred as a number somewhere around like 0.2, 0.8, 1, or 1 point something if it is a very high powered objective lens. And it is proportional to the size of the lens and the focal length as well. Another important parameter is how wide of a field of view you can see with that. It is the field of view, and it's usually next to the magnification of the objective lens that you're using. A field number is noted that's related to that number. And usually when you are imaging a field of view, you trade off between field of view and magnification. So there is a, if you want to see bigger magnifications, like more detail, you need to sacrifice the field of view. So there's a trade-off, but there are unconventional approaches. I'm not going to go into detail, but I just want to put a note. If you need to, for instance, monitor microsieverts in milliliters of liquid, like hundreds of them together, and you want to do with a decent resolution. Is there a solution? Yes, actually, there are methods which are like holographic methods and on-chip imaging methods. They do not use lenses from time to time, but they can sometimes provide like a bit unorthodox solutions to these type of imaging problems that you may have, just as a side note. Another important, sorry, one back, please. I'll just, yeah. And then another important one is depth of field and resolution. So what is the diffraction limit in our system? So that is actually the best that you can do with your system. So when you have everything perfect, you focus, and then that is the smallest spot that you can achieve. It is a physical limit. It is something around half of the wavelength that you can achieve it. Next, please. So if you want to see so if you're imaging with 500 nanometer wavelength, and you want to see something 50 nanometer, so half of it is 250. So what is it for you? What is the solution for you? There are options to break diffraction limit. We call them super resolution microscopy approaches or optical nanoscopy methods. So they are some additional information or side shortcuts to resolve even smaller features, like yeah, as we said, some tubular structure, like cellular structures and stuff. So it was 2014 chemistry Nobel Prize was given to Stead, Storm, and Palm. These were some of them. Some of them are stochastic, some of them are deterministic. So there are options for you. If you can move forward, thank you. So if you are limited to diffraction limited domain, and you are interested in features somewhere around the micron reasonable range, what do you do? So most of the time imaging is a signal to background ratio game. So if you have infinite signal to noise ratio, you can resolve almost everything. So there is a lot of effort going to reducing the background. So when you have a wide field imaging setup, usually scenario happens, you shine light, you excite everything, you illuminate everything, and then you try to focus and image what you're interested in. But when you excite everything, when you illuminate everything, there's a lot of background. So you see what you want to image, what you focus, but you also see a lot of things that you don't want to see, and that is contributing to your noise floor, right? So there are ways to minimize that. For instance, you can limit the excitation, the portion of the sample that you are eliminating by using, for instance, total internal reflection. It can work with cells that are sticking to cover slips, but pretty much for that. Or you can use other techniques like confocal microscopy, multi-photon microscopy, and you can minimize your background and improve your axial sectioning. If we can click forward. For instance, here you can see, like, when you do wide field versus confocal, there's a huge, you see huge contrast improvement, and there's a sharper detail. One more, please. So I want to give a few examples. What are the working mechanism of laser scanning fluorescence microscopy methods? For instance, in confocal, the method that we use to minimize the background and provide axial screening is to do spatial filtering. We use two pinholes, one in front of the detector, the other one is after the laser. So we arrange it in a way that we only detect light coming from the focal in focus plane, and everything else, other planes, they will hit to the block and they will not reach the detector. So this allows us to limit the background, so it will result crisper images. And another way, if you think that this is too slow for you, very inefficient in terms of photon budget, so you can excite with a sheet. So we call it light sheet microscopy. The way that you suppress your background is, so when you excite in one dimension and in imaged and detect in the same dimension, it is hard to block the background. But if you separate these two arms orthogonal to each other, 90 degrees, so you actually almost automatically get rid of the background easily, right? So this allows you to suppress and also speed up the process. So you excite your sheet, and this will help you to speed up imaging process like 100 times, 1,000 times easily. So another approach for this is multi-photon microscopy. So in confocal microscopy, we already talk about luminescence, right? We excite and we emit a lower energy photon, but you can play a trick here. Instead of using single photon, you can use 2-photon of half energy. This will allow you to excite same energy level, and you're gonna emit almost same emission. So this, how will this benefit you? If we can click one more, please. So if you see the dye volume on the left, so when you do single photon excitation, you excite a lot of background on the top, because to reach to your focal point, you also excite everything on your way. But when you want to hit with 2 photons at the same time, this is probabilistically a low probable event. So you have to put a lot of flux, and this only happens at the focus where the flux is highest. So this automatically actually filters out all the background that you would cause if you were to excite it with single photon. Can we move forward, please? So the advantage is we automatically get the filtering, and since we operate lower energy photons, they're higher wavelength, we can easily penetrate even deeper. So that's great. So if it is a great thing, why don't we go 3 photons, 4 photons, 5 photons, right? We can do that. And actually, there is a 3-photon approach to it. You can even image deeper. Here, the graph compares 2-photon microscopy in pink and blue in 3-photon microscopy, how deeper you can go. Like, we can reach white matter to a level with 2-photon microscopy in brain imaging, but with 3-photon, we can even go subcortical regions. So here I would like to actually announce that we already purchased a 3-photon laser, and within a matter of weeks, we hope to have a functioning 3-photon microscope in our optics division. So a lot of opportunities there. So it is, I think, a great news. We are very excited about that. So if we can click more, so is it always better to go more photons? I mean, it's great. It has a lot of benefits. However, it is not always the best approach. So if you want to image something shallow, it is not good to go with 3 photons, because there are also risks coming with it. So you can heat up your tissue a lot, because you can tell, right? It is even less probable event. So you need to pump a lot of light to that point, right? So there is water absorption increases as you go that wavelength. That's another problem, and you can induce temperature increase. So there are some cool tricks that you can use different wavelengths of photons, and you can combine them, and you can even get some benefits of 2-photon and 3-photon in a cool way. So what are other, so sometimes you need special needs, right? When we do laser scanning microscope, the shape of the beam is Gaussian. So in X, Y, and Z, you have almost a uniform shape point spread function. That's how we define PSF. So PSF is how one point is observed in your system. So sometimes this is not the best method for you, because if you are looking into a sample that is a bit sparse, and you want to be able to see all the depth together, you don't want to see like micron slices. You want to see 50 micron altogether, because they're not overlapping already too much. This way, you want to speed up things, and maybe your sample is moving up and down in Z, because it is breathing or some other reason, right? So you want to minimize this focal volume going in and out of your focus. So this can be a very cool approach to use pencil-like beams. They're called bezel beams. So what we can do is we can use tiny array of mirrors to shape our wavefront. And this provides us multiple benefits. We can shape our point spread function in the way that we want. Also, we can use these tools to further correct some wavefront problems. Sometimes things are not perfect, right? Optics is not perfect. There are other aberrations coming from your system. For instance, if you're imaging retina, there's a lens in front of it. So you have to correct some of these curves. So this actually tools can also help you to adaptively correct your wavefront. So these are very impressive and useful tools. We can move forward. So I want to touch some aspects of light and how we are using them to image some aspects. So light is a wave, and it has coherence properties. So this means as single color as light is, like lasers or maybe LEDs, they are more and more coherent, meaning that these wavefront are aligned. It has a well-defined relationship in phase from different points in space. I mean, we can discuss coherence in spatial and temporal, but I'm not gonna just diverse it into. So since we have this property, so we can, so since due to these properties, light can actually interfere constructively or destructively, and we are using them in some of our imaging methods. If you can move forward. Yeah. For instance, optical coherence tomography is a modality, optical imaging modality that we are very widely using, which relies on coherence properties, and it's an interferometric detection. So we have a light source, low coherence, we split it into two. One of them interacts with sample, the other one goes to a reference, and it comes back and it interferes with each other. Depending on the difference caused by the sample, we can actually get a lot of properties from the sample. It is a very, so it is, you can think of as light equivalent of ultrasound imaging, if you're familiar with that. So since you have phase information, you can go Doppler information as well. It is very important, it is very useful tool to image wide areas. So it can go as deep, a bit more even deeper than millimeter. So you can look into flows, biological flows. You can look into vascular structure or skin or others. So it has already made its way to clinic with eye imaging. It's very commonly used ophthalmology. Can we move forward? So we use optic, as I mentioned. I will cruise through these applications just to give a sense what we do with them. So we are using OCT to image blood flow, morphology, and we also use the polarization sensitive OCT to do post-mortem human brain imaging as well. So it gives us the orientation. So next, please. Yeah, coherence gives rise. So if the surface that it interacts with is granular, we end up with the speckle feature, which is a result of coherence of the light again. So this is useful actually. It doesn't look like much, but it can be, it can contain a lot of information about the sample. One more, please. So if we shine coherent light to a surface of the tissue, in A, you see this granular speckle, right? So, but this speckle, the temporal dynamics of speckle is dependent on whatever is moving underneath. So if these scatterers, we call them scatterers, are moving faster versus they're static, they will have a different dynamic. So by studying this, we can actually construct relative blood flow maps. So we can have nice vascular maps of the tissue underneath, as you can see here. So it gives us relative measurement of cerebral blood flow. Yes, next, please. So combining this with, so we can also use absorption properties together with this speckle contrast by illuminating with different colors of light. We know like these molecules have very specific fingerprint-like structures, oxygenated versus deoxygenated hemoglobin, for instance. So by measuring at different points, we can calculate the oxygenation accurately. So by also knowing the cerebral blood flow from laser speckle, we can combine these two and calculate cerebral metabolic rate of oxygen. Next, please. So we also use these laser speckle contrast imaging for different applications. Like this is after stroke. We use this to analyze penumbra size. We can, so this is a macroscopic approach. So you can image millimeters. So what you see here is whole mouse brain over skull. So we can study this penumbra size, and we were able to compare them depending on which circadian rhythm they experienced the stroke at. Next, please. this is another example where this optical intrinsic imaging, this absorption characteristics, and flow together was analyzed again in a stroke example. So I'll pass through them, cruise through them, and I will be happy if there are questions. So another approach that we use is two-photon microscopy, and we combine this with OCT, optical coherence tomography that I already mentioned, and intrinsic imaging, which is already mentioned. So we have this multimodal platform. We are using these to image brain in awake animals, behaving animals. So we have a probe. We are using luminescence in this case. We inject vascular or into tissue, and that is sensitive to the PO2, partial pressure of oxygen in the environment, in microenvironment. And the lifetime of phosphorescence is depending on how much oxygen in there, and it is very well calibrated. We can quantify the oxygenation very accurately, absolute values in behaving behaving animals. And we can also look into how it changes as a function of functional activation. In this case, it was whisker puff. And we can also see as a side product, we can see RBC passages because the intensity flux, the dye is staying in the plasma. So whenever there is a cell is passing through your focal volume, it will cast a shadow and you will see these blips, which are gonna form the red blood cell flux ratio. And we can perform flow and oxygenation measurements together, so we can see the dynamics and how do they interact with each other time-wise as well. So we can move forward. So brain is not the only organ that we can do that. Recently, over the last few years, we also did imaging in retina as well, through pupil, intact retina, non-invasively. So we were able to quantify PO2 in absolute terms in mouse retina at 3 different layers by using this method as well. More, next, please. So in this structure, it was depth dependent. So it was the first time that it was demonstrated in this manner. So that was quite opening many possibility opening application. Also, another organ that we recently reached to is intestine. So we are measuring the oxygenation in microvasculature in mouse cecum. It is in the large intestine side. So we can study the structure, flux, and oxygenation as well in subcapillary level. Next, please. So with that, I will pass the ball to Bill, and thank you for your patience, and thank you all the contributors, collaborators, and funding agencies.
Thank you so much, Ikbal. We'll now switch to Ben. I suppose I have one question while Ben switches. And so how do these, so you covered like a huge range of methodologies. So how do you validate whether these are actually measuring the right thing? Are there other imaging techniques that you compare optical techniques to?
That's a great question. So it is like being a responsible user actually requires knowing exactly what you measure. For instance, simply shining light that is within the range into hemodynamics, so absorptive range, you may think that you're measuring one thing, but you may be measuring something totally different. Yeah. So being aware of the dynamics of the tissue, I think Bin is going to give a lot of insights in that regard, is very important. And whether or not optical measurements are always the best, sometimes it's not. Sometimes you don't have to see all these structures, but if you need some spatial information, how these features, how these structures in 3 dimension are talking to each other, how they are located, sometimes you need that location, spatial information in addition to the values that you measure. Exactly. So sometimes in different modality may be better for your applications, but optics is a wide area, so you will find one for your needs, I would say.
All right. Thank you so much.
Thank you.
Thank you so much for the nice talk. And so we'll now move on to Ben. And yeah.
Can you hear me?
Hey, Ben. Yes, we can hear you.
Okay, great. So that was a very impressive talk. And now I'm gonna change gear to translational biomedical optics. And I'm quickly gonna go through some of the underlying operating principles and introduce some of the tools we use to do those clinical studies, and then give you some examples of the current project we're doing. So I want to bring back to this diagram Ikbal showed earlier. So she has covered most of the high resolution imaging modalities that shows you crispy result and with high resolution structures. But now when we move to NIRS and DCS, things will get blurry, and some of the time we don't see the image at all. It's mostly a point measurement of the tissue physiology. But the benefit of NIRS and DCS is that it can go deep into the tissue, and that is very appealing to clinical applications. So I'm sure many of you have tried this trick when you're young, like you put your finger or head against or face against a flashlight, and what you observe is the hand is glowing red. But why do we see red when the flashlight was white? So the answer is that red and the invisible near-infrared light actually can penetrate through the tissue, while the rest of the spectral component get blocked in the meantime. And the other thing you notice that the glow is much bigger than the area of the flashlight. So that means the light is not penetrating through the tissue in a straight walk. And that's exactly what happens in tissue. So in tissue, there are two events that dominance. One is absorption and one is scattering. So whenever the light interact with the scatterer once in the tissue, for example, cell nuclei, mitochondria, or whole cells like red blood cells, it changed trajectory and lose some energy in the meantime. And in tissue, the scattering coefficient, which is the inverse of the length light travels between two scattering events, is about 2 or 3 magnitude bigger than absorption. So that's why you see a zigzag move of light. And the way you imagine like how it travels in tissue is very much like a drop of ink in a glass of milk. And you see most of the forward motion and while it's spread out in the meantime. And as you can see that after a few scattering events, light actually loses its original direction. So if we make the assumption that the scattering event is isotropic, meaning it has equal probability in scattering in all directions, you can reduce the scattering coefficient to in the quantity called reduced scattering coefficient. That's easier to qualify. And in tissue, the reduced scattering coefficient is like 1 millimeter. That means light will completely lose their initial direction after 1 millimeter depth. So in the microscopy domain, which operate like a couple hundred microns, you can see the high contrast, high resolution image, whereas in the diffuse optic domain, it's all get blurry. And the slight propagation in tissue can be mathematically described by the radiated transfer equation. And basically it describes the conservation of energy of light propagating in media. It basically describes that at a certain location, once in the tissue, at a certain time, once in a certain direction, the radiation in that area, in that media, such as light source, plus the gain of light that got rescattered in this direction equals to the temporal and spatial variation plus a loss from scattering and absorption. And in human tissue, because the scattering coefficient is much, much bigger than absorption, and also when we use the technique, the source and detector separation is much bigger than the scattering length, we can make this diffusion approximation assumption and simplify the RTE equation to diffusion equation. And whereas the effective diffusion coefficient contains the absorption reduced scattering component allow us to calculate the tissue optical properties. And as Ikbal has shown this figure earlier, the reason light can penetrate deep into tissue in the near infrared light region is because 70% of the most abundant molecule water in human body is actually have a very low absorption in this so-called optical window that spans from 650 to 950 nanometers. And also even luckier, the oxygenated hemoglobin and deoxygenated hemoglobin has very different spectrum in this region too. And if you put one laser wavelength to the left of the isobastic point and one to the right, you can actually calculate delineate the oxygenated and deoxygenated components of hemoglobin concentration. And these are the biomarkers we identify using NIRS technique. And for the scattering part, the scattering coefficient follows a power law dependence on the wavelengths. And the power here is something of physiological relevance that can be used for as a biomarker. So before we get into the techniques we use, I want to touch base on the sources and detectors, and also how to move light around to deliver the light into human tissue. So on the source side, we have the lamps, which has broad spectral excitation range. And then we have a very cheap option of single wavelength LEDs. They're very robust, but doesn't have very high spectral purity, and it's not easy to work with fibers. However, they're very easy to work with if you're developing some portable systems that I'll show later. And the most of the light source that we use in our application is laser diode. It has high power and has better coherence length and stable, and it's easy to be coupled with fibers. However, they're pretty delicate and easy to bend down, and they're more expensive. And the most fancy sort of light source are the laser systems that can range somewhere from a single laser diode connecting with the cavity to stabilize the laser power to a desktop system that can give you femtosecond narrow laser pulses and super high powers. But of course, they're more expensive. On the detector side, the power horse is the photodiodes. Basically, it's silicon-based semiconductors that use the PNA structure to convert light into current. It's relatively cheap, and it has pretty good sensitivity in the near infrared range. And if you want higher gain and higher sensitivity in applications where the light is power is pretty low, you want to look into the avalanche photodiodes or APDs. However, they're more delicate and a little bit more expensive. And the third one is photomultiplier tubes, and those are vacuum phototubes. Basically, work similarly like the avalanche. It has the mechanism to induce the cascade event to amplify the laser power, to amplify the signal level, and it can reach 100 million multiplications in to amplify the signal. However, it requires pretty high voltage and prone to damage. And the most important part is PMTs has very limited sensitivity to near infrared light, whereas the silicon counterparts can go as high as over like 1000 nanometers. And also PMTs are very sensitive to magnetic field, and depending on if you want to use it together with MR scanner, it's not going to work. So the thing we use to connect the source and detectors are frequently the fibers. So it's composed of a silica core and with some glass claddings, and they have some coatings color coded to differentiate the mode of fiber. And usually you put some jacket and those fibers to help them maintain the shape. And there are single mode and multi-mode fibers, and the most dramatic difference is the diameter of the intercore. And the intercore of the single mode fiber is only 9 micrometers, very small, whereas the multi-mode can have a core diameter from 5 to 400 microns. It allows to gather more light to deliver to the tissue. However, because it can allow multiple mode of total reflection inside the fiber, the more times that the total reflection happens, the more probability the light get attenuated. So it attenuates fast. And but the cost of the multi-mode fiber is like the half or less than a single mode fiber. And so use the fiber optics, we can use one source and multiple detectors and put them together in the so-called probe that you can put onto tissue, into contact with the tissue. And the sources, you can combine multiple fibers, even with multiple modes, to do multimodal applications. So those I mentioned earlier was mostly for contact optical imaging applications, and there's non-contact options that you can use CCD cameras as the detectors and a digital mirror device as the source to project multiple different patterns on the tissue and get the optical properties that way, such as in the spatial frequency domain imaging. And today I'm not going to talk about this technique, but it's possible to do non-contact optical imaging. So the technique I'm going to talk about today is NIRS, the near-infrared spectroscopy, and diffuse optical tomography, and diffuse correlation spectroscopy. So first, let's talk about NIRS. So if you put a near-infrared laser source here and put a detector here, and this basically shows in the 3D spatial sensitivity profile of what the detector can detect. Basically, this shows the volume of tissue this source and detector pair is gonna sample through the tissue. And there are multiple ways to do the work. One is to shed lasers with continuous light intensity, and you can measure as the laser go past the tissue, the intensity gets attenuated, and you can measure difference. And from this difference, you can calculate total attenuation, but you can't differentiate the absorption and scattering events apart. So this is not ideal, but if you only care about relative changes of hemoglobin, this is good enough. And if you want the ability to tell those scattering and absorption event apart, you can use the radio frequency to modulate the light. And as the modulated light passes through the brain, past the tissue, not only it get attenuation, also get the phase shift of the modulated light. And from this diagram, there are two unknowns and two pieces of information. You can basically resolve the absolute values, and that will allow you to direct quantification of hemoglobin concentration. And this is mostly applied in cases where the absolute values is relevant. And the third method is called time domain NIRS, where you shed a very narrow light pulse into the tissue. And because the Fourier transfer tells you like a very narrow band light contains multiple frequencies, so this modality contains the most informative information about tissue properties. And one appealing trait about this technique is that the absolute values, the absolute intensity of the detected light is no longer relevant, but the slope of the temporal point spread function carries most of the information of the scattering, and the shift of the peak from the TPS as per curve from the instrument response function tells you mostly about the scattering. So when this absolute intensity doesn't care, that means if there is some change of light intensity caused by motion, it's not sensitive to that. So that's very, very neat. But this is also the most expensive method. So I'm going to give you an example about the application of NIRS technique, and the most popular, one of the most popular application is called functional NIRS, and basically it measures the relative hemoglobin changes due to functional neural stimulation, and it's the same idea as both fMRI, but we done it with the optical method. So as you probably all know, both is sensitive to HBO concentration changes, whereas with NIRS, by using multiple wavelengths, you can delineate HBO, HBr, and can calculate the total hemoglobin concentration. And the optical method is relatively, it's more accessible than MRI. Actually, there are several commercial companies already manufacture FNIR systems that you can use. And because the NIRS, you can wear it as a cap, and it's less sensitive to motion. So you can do functional tasks on babies and infants who is less likely to stay still in an MR scanner. However, this one, the FNIRS can only reach to the deep depths of cortex, and it's not a whole brain imaging modality, and lacks of anatomical references that MR technique can provide simultaneously. And this because if you remember clear, as I mentioned earlier, because the light samples a big volume of the brain, it tends to get systematic physiological crosstalk from the superlayer blood flow signals here. And however, recently we've developed some technique by using a short separation detector to sample only the superficial layer, and you can regress the influence of the systematic physiological crosstalk. So this is an early study that basically did the simultaneous NIRS and the both imaging, functional imaging together. And you can see nicely that the NIRS HBR response, so inverse proportion to the both signal very well. And if we normalize the signals to the baseline, you can see they overlap like nicely. And also you can see there's delayed response compared to the HBR, HBT that basically, that basically is the dilation of the flow change that happens before the both signal. And this is a work done by Maria Angela Francini at the Optics at Martinos. And here we did the typical finger typing experiment, and you can find the classic response of a hemodynamic response due to the functional activation. And this is if you put a probe that in a certain brain region, and if you wear a cap like this that cover multiple regions of the brain, you can basically do the similar resting state functional connectivity using optical method, similarly as the MRI scans. And this is just the example of several studies that shows the contralateral connectivity between the left and right hemisphere. And I want to highlight this study to the lower right corner is that this is a longitudinal monitoring study on infants from neonates to 3 and 6 months old infants. So once NIRS, you can possibly do brain developmental imaging studies like this. And the future trend of FNIRS is wearable devices like this. There are several commercial companies doing their modularized the source and the NIRS modules and make it wearable. And there's also a Society for Functional Near Infrared Spectroscopy, which is founded in 2014 by the center's David Bowles. He moved to BU now, but so far, as of today, there are 1.6K members on Facebook, and there have very active talks about functional NIRS. Sorry, there's typo. It should be functional NIRS. So functional NIRS applications, if you're interested, you can follow those. So the talk before talking about like only maybe one source and several detector distances. But if you put an array of sources like this on a plate and another array of detectors on the opposite plate, you can basically do the 3D tomographic imaging of human tissue. And here we use a combined CW and the frequency domain NIRS system to measure, to do the optical breast imaging. And we combined the DOT with the X-ray digital tomography. Similarly as like PET and CT, one offers the anatomical structure and one offers the functional differences. And this is actually a clinical medical device we use in clinical space. So we enclose the device, enclose the tower so people don't get scared of all those 174 fibers. So this is just a case showing what the contrast we see on breast cancer patients. So one of the markers that get consensus in the field that shows tumor differences is total hemoglobin concentration. And in this case, you can see the elevated HBT in the tumor region. And because solid tumor is stiffer than the normal tissue, if we apply stepwise pressure, the solid tumor is more likely to bear the compression and squeeze out more blood content out. So if you subtract the optical image at full compression from the half, you can see a decrease of the marked region even better. And the time course also show a dramatic difference between the tumor and normal tissue area. So this has the clinical relevance of differential diagnosis of malignant and benign tumors. So that is for the diagnosis part of the application. And there's also clinical opportunity that is to monitor the therapy monitoring response. So for women with diagnosis of locally advanced breast tumor, usually they'll get the so-called neoadjuvant chemotherapy. So that happens before they get the surgery to remove the lesion. And this therapy usually lasts 3 to 6 months long. And the clinical tool to evaluate the response to the therapy is surgically removed breast tissue, which obviously too late by the time you get the surgery. And also the therapy, the neoadjuvant therapy has a very heterogeneous response. Not all women respond to the treatment. So it's actually lose a window of opportunity to change the treatment regimen if the patient is not responding. So optical imaging offers an alternative tool to trying to define the early signs of response. So people who are not responding can be switched to a more effective treatment. So our study has shown that between at the baseline, the responder and non-responders have similar trend where the tumor shows a decrease, a more dramatic change upon compression. And at 30 days, the changes stays pretty much, the changes kind of getting smaller for responders, whereas for non-responders, the changes has been, the effect has been more prominent. And thus differences in the normal and tumor tissue response under compression can potentially be a marker for therapy monitoring result. So now I'm gonna change, I think it's pretty long. It's kind of over time. So this is the last technique I'm gonna talk about, so bear with me. The diffuse correlation spectroscopy is a very different technique than the NIRS, and they're complementary. So basically, the physics behind it is very close to what Ikbal has described in the laser speckle imaging. So basically, you shed a coherent light into tissue, and here we got lucky because the scattered ones in the tissue is the red blood cells, and we have sensitivity to red blood cells, and they're also very relevant to the tissue physiology. And by shedding a long coherent light into tissue and interact with the moving red blood cells, you can use a single photon counting detector to log the time traces of the laser, the detected light intensity, and correlate with the instant light. And you can define this temporal intensity autocorrelation function by using this equation and give you this so-called G2 curve. So that means once at millisecond time frame, the laser is still in good correlation with the original instant light. However, at the millisecond time, they lost correlation and completely like ran into noise. And something in between tells you how fast the red blood cell moves. And this has been modeled using the equation here, and this moving, dynamic light imaging part give you a quantitative measure of blood flow index. And on this G2 curve, faster flows move the curve to the left, and you can use the fitting to figure out the blood flow index. And what makes it powerful is the combination of DCS and NIRS. It can offer a measurement of the cerebral metabolic rate of oxygen. So basically, the cerebral metabolic rate of oxygen can be described using this equation here. And the DCS can measure the blood flow index, whereas the NIRS can measure the oxygen saturation within the tissue. And together, they can allow the measurement of cerebral metabolic rate of oxygen. And CRML2 is fundamentally related to the tissue health, especially for the metabolism-dependent tissues such as brain. And in this example, I talk about using NIRS and DCS to monitor the brain health during cardiac pulmonary bypass. And this is the project done by multiple PIs at the Optics at Martinos, Dr. Stefan Kapp, Dr. Mariangela Franschini, and Dr. Jason Chi from the MGH anesthesia department. So cardiopulmonary bypass is used for cardiac surgeries that need heart motion to be stopped and then connect to an external bypass. And then under anesthesia, the oxygen supply to brain is impaired due to the reduced blood pressure. And while it's on bypass, the cerebral perfusion is maintained by indirectly by controlling the arterial blood pressure to be within the limit of autocorrelation, so that the human body can regulate the blood flow. And currently, however, there's no clinical approach to monitor the cerebral blood flow directly for a long period of time. And in this ongoing investigation of brain health during CBP, we use combined DCS and NIRS measurement to allow us to measure the blood flow and the fMRI together. And we have a multimodal probe that is placed on the forehead of the patient. And this is an example result of a patient bypass surgery. We monitor the whole thing like during the entire bypass surgery. And the common feature, the typical feature we saw in all subjects is that the total blood, total hemoglobin concentration show the top right panel dropped when the patient moved on to the bypass because the blood is thin to reduce the chance of clotting. And they partially recovered once they're moved off bypass and back to normal. And the other thing that is the blood flow index and the CRML2 are very well correlated with each other. And these things we observe is that during the rewarm phase or in the procedure, they both progressively increase because temperature increase motivate the increase of blood flow, and the chemical reaction on the tissue kind of accelerate the oxygen consumption during the process. In this study, we noticed there's one case that shows this interesting oscillation. It's a very low frequency, and it happens at the like minute scale, whereas the anterior blood pressure, whereas the blood flow index varies together with the anterior blood pressure, which is not supposed to happen because the tissue the autoimmune regulation is supposed to compensate for the flow to maintain stable. So this basically indicate of impaired auto regulation, and this correlation between this blood flow index and the arterial pressure only happens during the bypass periods. So this indicates something serious and need to be paid attention to. So this is still a very early investigation, and we're still trying to make sense what to read about this result. And it's a very important study that is addressing an unmet clinical need. So with that, I'd like to thank you all the people at the optics at Martinos, and also our collaborators and at the Department of Radiology, Cancer Center, and Anesthesiology. Thank you, and sorry for the disconnection.
Thanks a lot, Ben. That was a great talk, an impressive sort of range of techniques being covered. So yeah, so the next one now is going to be on fMRI, and Kaiser will be presenting it next week. So please look out for the email. If there are no more questions, I don't see any questions. So we'll end the talk now, and you can see the recording on YouTube. All right, thanks a lot, Ben. Thanks a lot, Ikbal. Bye-bye, everyone. Bye-bye.
Thank you. Bye.
Bye.